Apoptosis. So apoptosis is a type of programmed cell death that is induced by a tightly regulated intracellular program. It is characterized by the activation of intrinsic enzymes of the cell that degrade its own nuclear DNA and proteins. So fragments of the apoptotic cells break and fall off from the cells, and it is hence named apoptosis. Ptosis means falling off, okay? So causes of apoptosis, physiological situations in embryogenesis, we can see removal of the excess cells such as disappearance of the web spaces between tissues and fingers. And again, the elimination of cells after withdrawal of the hormonal stimuli, for example, endometrial shedding during the menstrual cycle, and then elimination of cells after withdrawal of tropic stimuli, for example, when neutrophils in an acute inflammatory response after the inflammation is done, they apoptose. And again, elimination of potentially harmful cells such as self-reactive lymphocytes. Pathological conditions, we can see apoptosis whenever there is DNA damage. So any drugs, radiation, whenever that causes DNA damage, there is P53 triggered apoptosis. So mainly tumor suppressor gene P53, it recognizes cells with damaged DNA and assesses whether the cell can be repaired or not. If the damage is too severe, P53 triggers apoptosis. Next, it destroys cells with dangerous mutations or with DNA damage, so apoptosis. Because if it is beyond repair, then apoptosis prevents the development of cancer. Next, elimination of cells with excessively accumulated misfolded proteins. So mutations in the genes encoding proteins or extrinsic factors may result in accumulation of unfolded or misfolded proteins. So excessive intracellular accumulation of these abnormally folded proteins in the ER is known as ER stress. It results in apoptotic cell death. So excessive proteins, excessive DNA damage, and a killing of viral infected cells. So in viral infections, the infected cells are lost mainly due to apoptosis induced either by the virus or by the host human response by cytotoxic T lymphocytes, as in viral hepatitis. And again, elimination of neoplastic cells or rejection of the transplant. So the T cytotoxic cell mediated mechanism is responsible for apoptosis in tumors and cell rejection of transplants. And again, elimination of parenchymal cells and pathological atrophy. So obstruction of the duct in the parenchymal organs like pancreas, it can also lead to apoptosis. Now, morphologically, you can see cell shrinkage. See, everything shrinks and everything spreads out. Apoptosis falls off, okay? So cell shrinkage, the cytoplasm becomes dense. So as you can see here, the cytoplasm becomes dense. Then there is nuclear condensation and fragmentation. What happens is chromatin, it condenses in the periphery of the nuclear membrane. Chromatin aggregates in the periphery. You can see the darkness here, okay? And then it aggregates in the periphery, and the nucleus may break into two or more nuclear fragments. So the nucleus, it breaks down, okay? The nucleus breaks down. Next, the cytoplasm also breaks down. So there is formation of cytoplasmic blebs and also apoptotic bodies. First, the cell shows extensive surface blebbing, okay, followed by fragmentation of the blebs into membrane-bound apoptotic bodies. They are composed of cytoplasm and tightly packed organelles with or without nuclear fragments, okay? Next, so once these apoptotic bodies are shed out, then they are phagocytosed by the macrophages. So phagocytosis of the apoptotic cells or bodies is usually done by the macrophages, and it is degraded by the lysosomal enzymes of the phagocytes. So light microscopic changes, you can see an intense eosinophilic cytoplasm, and the nuclei appear as fragments of dense nuclear chromatin, and there is pycnosis, and apoptosis does not elicit an inflammatory reaction in the host, okay? There is no inflammation associated with apoptosis in the host. So mechanisms of apoptosis, it is regulated by the biochemical pathway. There are two opposite sets of signals, namely death signal, pro-apoptotic, and anti-apoptotic or pro-survival signals. Unlike necrosis, apoptosis engages the cell's own signaling cascades and results in its own death. So apoptosis, it results from activation of enzymes called caspases. They are cysteine proteases that cleave the proteins after aspartic residues. Cysteine, they cleave after aspartate. Next phases are initiation phase and execution phase. Initiation phase. So apoptosis is initiated by signals derived from two distinct pathways, the intrinsic or mitochondrial pathway and the extrinsic or death receptor pathway. So in intrinsic or mitochondrial pathway, what happens is mitochondrial damage is a major mechanism. So what happens is their mitochondrial membrane permeability, it is controlled by BCL2 family, B cell lymphoma family, okay? So BCL family consists of pro-apoptotic proteins like BAX and BAC and anti-apoptotic proteins like BCL2, BCLXL and MCL1. These anti-apoptotic proteins, they prevent leakage of mitochondrial proteins such as cytochrome C, okay? Mitochondria, they contain several proteins like cytochrome C and pro-apoptotic proteins. Once they leak out, they cause apoptosis. So these anti-apoptotic proteins BCL2, BCLXL, MCL1, they prevent this leakage. So growth factors and other survival signals stimulate production of anti-apoptotic proteins. If the balance shifts to pro-apoptotic proteins, then the apoptotic cascade is activated, okay? Now, causes of mitochondrial injury. The pro-apoptotic signals include deprivation of the growth factor or DNA damage, any accumulation of excessive misfolded proteins or ER stress, and increased intracellular free calcium. So steps in the intrinsic pathway, what happens is mitochondrial injury causes increased mitochondrial permeability and release of pro-apoptotic molecules into the cytoplasm. So what happens is the above mentioned causes of mitochondrial injury, they activate a number of sensors of the BCL2 family. BCL2 sensors, they are called BH3 proteins. So has BCL2 homology. So homological B proteins, they activate two critical apoptotic BCL2 family effector proteins, BAX and BAC. They create channels in the mitochondria. They increase the permeability of the mitochondrial membrane and allow several mitochondrial proteins, pro-apoptotic ones such as cytochrome C, to leak out from the inner mitochondrial membrane into the cytoplasm. So cytochrome C, it is released, it binds to APAF1. Cytochrome C binds to APAF1, which is apoptosis activating factor 1, and it forms an apoptosome. Apoptosome binds caspase 9, which is a critical initiator caspase of the mitochondrial pathway, which sets in an auto-amplification process. Okay, the cytoplasm of the normal cells contains proteins which block the activation of caspases and function as physical PIs or IAPs, physiological inhibitors of apoptosis. So outer mitochondrial proteins may alter the cytoplasm and they neutralize these IAPs and initiate the caspase cascade. Next, sensors of BCL2 family, namely BH3 only proteins, they also bind to and block the function of protective anti-apoptotic proteins like BCL2 and BCL-XL. Okay, that is what happens. And in the extrinsic death receptor mediated pathway, what happens is many cells express death receptor molecules on the surface of the plasma membrane that trigger apoptosis. So death receptors, they are a member of the tumor necrosis factor family. They have a death domain or a cytoplasmic domain. So if they are located in the plasma membrane, they have a death domain or cytoplasmic death domain. They are members of the TNF family. And they also have a related protein called FAS. Okay, FAS. FAS is also called TNF-R1 and CD95. Okay, so they are members of the TNF family and they contain a death domain in the cytoplasm, and apoptosis is initiated when FAS ligand present on the bacterial cells or self-reactive T lymphocytes binds to the viral cells or self-reactive lymphocytes binds to the FAS ligand. FAS ligand binds to FAS, and then the death receptor, it is activated. Okay, so then what happens is self-reactive lymphocytes, virus infected cells and tumor cells, they have this FAS ligand. So the extrinsic pathway is activated when CD95 or FAS binds to FAS ligand. Then the cytoplasmic death domain binds with an adapter protein called FAD, FADD. Okay, FAD adapter binds to the death domain. Then FAD in turn binds to procaspase 8 and activates it to generate active caspase 8. Here it was caspase 9 and now it is caspase 8. So activated caspase 8 mediates the execution phase of apoptosis. Okay. Now in the execution phase of apoptosis, what happens is caspase 9 and caspase 8, they further activate executioner caspases. So caspases 3 and caspases 6. So what they do is they act on many cellular components and they activate DNAs, which induces fragmentation of the nuclei. And caspases also degrade components of the nuclear matrix and cytoskeleton, resulting in fragmentation of involved cells. Now the removal of apoptotic cells, it is usually by phagocytosis. So apoptotic cells in bodies are engulfed and removed by mainly macrophages. This is so efficient that these dead cells and apoptotic bodies disappear within minutes. Okay, there is very little chance of inflammation. So factors favoring phagocytosis. So again, apoptotic cells and apoptotic bodies, they undergo several changes in their membranes and produce signals that favor phagocytosis. So what are they? Phosphatidylserine expression. So usually it is present on the inner side of the membrane, phosphatidylserine in healthy cells, but in damaged cells or cells undergoing apoptosis, it turns out. It is expressed on the outer layer of the membrane and it is for phagocytes to recognize it easily. And secretion of soluble factors. Apoptotic cells, they secrete soluble factors like thrombospondin that recruit the phagocytes, and natural antibodies and proteins of the complement system, they may coat the apoptotic bodies, which aids in phagocytosis. So diagnosis or detection of apoptosis, you can do DNA fragmentation assay by electrophoresis. Apoptosis, you can see a stepladder pattern in contrast to smeared pattern in necrosis. And you can use tunnel technique. So terminal deoxynucleotide transferase, biotin, NIC and labeling. So tunnel technique for in vivo detection of apoptosis. And again, you can see chromatin condensation by H&E staining, and again estimation of cytosolic cytochrome C and activated caspases, annexin 5. Okay, that is because apoptotic cells, they express phosphatidylserine on the outer layer of the plasma membrane, and these are recognized by the diannexin. And also propedium iodide assay by flow cytometry. Next, disorders associated with dysregulated apoptosis. You can have cancer or autoimmune diseases. Disorders with increased apoptosis, you can have neurodegenerative diseases like Alzheimer's. There is accumulation of improper protein, Huntington, Parkinson disease, ischemic injury in MI and stroke, and again death of virus infected cells in AIDS. Clinical significance of apoptosis in cancers. Normally, cells with damaged DNA are cleared in the body by undergoing apoptosis, but apoptosis may be reduced in some cancers. Best established role in protecting tumor cells from undergoing apoptosis is seen in follicular lymphoma. So in this type, you can see there is translocation of 14:18, which causes the overexpression of anti-apoptotic protein BCL2. So this in turn increases the BCL to BCL-XL buffer, protecting abnormal B lymphocytes from undergoing apoptosis.